Now we talk about proteins, which are other important components of life in addition to DNA and RNA. Proteins are made up of amino acids. Amino acids are the molecules that contains the amino groups, as well as they have some carboxyl groups, so they have both the acidic as well as the basic properties. Many proteins are enzymes, like they play the roles of like catalysts in the life processes, and they catalyze the chemical reactions in the cells. Yet there are other proteins that allow cells to move, regulate biological process, or function as structural components. So your muscles, they are made up of proteins. So the proteins are everywhere. So here we see in this diagram, and there is a structure of an amino acid. So amino acids are made up of an amino group, a carboxyl group attached to the same carbon. We call this carbon as alpha carbon. As you see, there is symbol over here. And then carbon has a valency of 4. So at the same time, it can attach to 4 different molecules. So down below, we see there is a hydrogen molecule. And up there is a variable group R. Just like in organic chemistry, you see there is something that can be a variable or something like alkyl group. We represent it as an R in that case. So that group is variable. So we can form different sort of amino acids when we see the variability in this group. Here are different amino acids. Amino acids there are 20 different types of amino acids. So they have been divided into different groups depending upon the properties which are there in those variable groups. So these are aliphatic R group containing nonpolar amino acids. As the name indicates, it's glycine right here, then alanine, proline, valine, leucine, isoleucine, and methionine. Then you can see there are 3 letter codes down there, and then you can also see their single letter codes. So those single letter codes are important for us. So when we talk about some amino acid chain or some protein, we will use these symbols. So here we see another group, which is nonpolar aromatic R group containing amino acids, which are phenylalanine, tyrosine, and tryptophan. Polar uncharged group amino acids, which is serine, threonine, cysteine, asparagine, and glutamine. Guys, since we have redundancy in the symbols, so we might have strange symbols, like you can see glycine, we have Q, maybe G is already taken by some other amino acid. We have polar positively charged groups, histidine, lysine, and arginine. We have another group where we have negatively charged polar R groups, which is aspartate and glutamate. So in total, we have 20 amino acids. So we can say protein is actually made up of 20 variables or 20 alphabets. So these 20, they can recur in any frequency and gives you many millions of the proteins. Here in this diagram, we see like how 2 amino acids, they combine with one another. So there is one amino acid coming, and that is the carboxyl group. We will see that this OH coming from one amino acid and attaches to the H coming from another amino acid. So there is a condensation, water moves out, and these two folks, they join one another. So this bond we call it as a peptide bond. So 2 amino acids, they get together with a peptide bond, and we find a protein molecule here. As far as the directionality of the protein is concerned, so since this bonding starts from this end, where on one side we have an amino group, we have a nitrogen, so we call this end of the protein as N-terminal, and where this chain ends, we would see this C double bond O, that is actually the carboxyl group. So actually these proteins, they have an N-terminal, and then they do have a C-terminal. Just like in DNA, we have a prime end and 3' end. So proteins they are designated as, their ends are designated as N-terminals and C-terminals. So they move from N to C. Okay guys, so let's see how can we define protein in a computing language. So protein, we see there are the variables, those 20 amino acids. So it's basically a character string which is derived from 20 variables or alphabets. Actually those amino acids, so A, G, C, whatever is it. So down below we see a protein structure which is actually in a FASTA format, where you can see this greater than sign, and there is some name of the protein, and then we have this structure which starts with M is methionine. So, and then obviously just like the DNA protein is also having those ends or terminals. So we can see this methionine is towards the N terminal, and this last amino acid is the C terminal of this protein. So this is how we represent those protein sequences. So later on, when we start playing with those sequences, you would see these sequences a lot.